Most human proteins have no usable drug-binding pocket. AI and new chemistry are finding ways around that constraint. Thursday, 8:10 p.m. A man sits on the edge of his bed, turning a pathology report soft at the folds. His doctors know the protein driving his tumor. They can trace the pathway. They still cannot point to an approved drug that reliably reaches it. That is not rare. By conventional small molecule rules, up to 85% of human proteins are still considered undruggable. The bottleneck is often not biology. It is geometry. Proteins are folded molecular machines, and standard drugs usually work by sitting inside a pocket, like a key in a lock. Many disease proteins have no pocket, no stable shape, or stay buried inside larger complexes. So labs turn to three tools: AlphaFold for structure prediction, cryo-EM for real molecular snapshots, and targeted protein degradation for removing proteins instead of blocking them. The real question is no longer whether we can name the target. It is whether structure prediction, molecular imaging, and new chemistry can convert a known disease protein into a medicine that works in patients. For decades, sequence data was not enough. Scientists could read the amino acid letters of a protein, but not reliably infer the three-dimensional fold that determines function. That folding problem resisted attack for more than 50 years. Then, AlphaFold changed the map. DeepMind system predicted many protein structures from sequence in minutes, often at near experimental accuracy. Reported performance reached a median score of 92.4 out of 100, and in some cases, sub-angstrom precision. By 2022, DeepMind and EMBL-EBI had released predictions for more than 200 million proteins. As Dr. Raminderpal Singh put it, this was less an AI wave than much more sophisticated mathematics applied to vast data. But a predicted structure is only a starting point. Cryo-electron microscopy solved a different problem. It flash freezes samples at about -183 degrees Celsius, then reconstructs proteins and complexes at near atomic resolution. That matters because many hard targets are not rigid. They flex, open, close, and interact with membranes or partner proteins. Cryo-EM can capture multiple states, including transient pockets that older methods often missed. Now the target is no longer just a sequence. It is a moving object with vulnerable moments. If 85% of proteins still resist drugs, stay here. CivilizationX is where we unpack the real constraints behind that number. And then the strategy changed again. Traditional inhibitors are occupancy-driven. They must keep sitting on the target to suppress it. Targeted protein degradation is event-driven. A degrader only needs to bring two proteins together long enough for the cell to act. But here is the hidden mechanism. One end binds the disease protein. The other recruits an E3 ligase, one of more than 600 enzymes in the ubiquitin proteasome system. The ligase tags the target with ubiquitin. The proteasome recognizes that tag and destroys the protein. Molecular glues do something even stranger. They are small molecules that create or stabilize a protein-protein interaction that did not usefully exist before, often pushing the target toward degradation. So the problem shifts. Instead of asking, can we block this surface? Researchers can ask, can we expose a hidden site, recruit the right ligase, and make the cell remove the protein itself? The search space is still brutal. Theoretical drug-like chemical space exceeds 10 to the power of 60 molecules, and existing AI systems have explored less than 10 to the power of -34% of it. Here is the constraint that stops the easy story. A model can predict a fold, but here is the problem. It cannot guarantee selectivity in living tissue. In a crowded cell, a degrader can bind a near-neighbor protein, recruit the wrong interaction, and delete something healthy. Success in silico can fail in blood, liver, or tumor. So, what have these tools actually achieved? They have turned many undruggable proteins from invisible objects into design problems. AlphaFold supplies candidate structures fast. Cryo-EM checks real conformations and complexes. Degraders and molecular glues create a path to catalytic, sometimes irreversible removal instead of temporary inhibition. But they do not erase failure. Drug development still often takes 10 to 15 years, can cost billions, and about 90% of clinical candidates still fail. The gap is no longer just finding targets. It is proving safety, delivery, and selectivity in patients. The next step is integration. Predict, visualize, perturb, then degrade. That stack is already pushing oncology, rare disease, and even cardiovascular target discovery, where AI, omics, and systems biology are expanding what counts as actionable biology. But what happens when the targets themselves seem out of reach? Some AI-native programs claim major cuts in early discovery time, and next-generation therapies, cell, gene, and nucleic acid treatments, are opening personalized options for chronic and rare diseases. But access is an engineering problem too. As Matthew Porteous argues, scalable, cost-effective medicine eventually requires factory-based industrialization. For the man on the bed, this does not mean the drug already exists. It means the sentence, "We know the target, but we still cannot hit it," is starting to change. Sometimes the target may not need to be hit. It may need to be exposed, redirected, or destroyed. Can researchers combine AI-predicted structures, cryo-EM snapshots, and the cell's 600+ E3 ligases to destroy one disease protein without touching its neighbors? The constraint is real. The data exists. Whether the systems built around it can act on it at scale, that is the next engineering problem. Stay with CivilizationX.